As an ambitious high schooler who wants to pursue a career in chemical engineering and a specialization in pharmaceuticals, performing and understanding what role titration plays is crucial. Titration is a process to determine the neutralization of an analyte and a titrant. Typically, an analyte is an acidic substance that scientists don't know the concentration of, while the titrant is usually a basic substance that we do know the concentration of. Neutralization means turning the pH of the analyte to 7, or also known as a neutral pH reading. This also means that there are equal amounts of base and acid in the analyte, and from there, scientists can calculate the concentration and sometimes purity of the analyte. But in order to follow any process, everyone has to know how to set it up. So here is a diagram of a titration, and as you can see in the burette, our substance is called the titrant, and the substance in the flask is called the analyte. And part of the analyte, or how scientists can determine that the pH is neutral, is the indicator. This indicator turns the clear analyte into a color, and in most high school titration labs, they use phenolphthalein, which turns the analyte pink. And just like a high school lab, I followed one that found the purity of baby aspirin. First, the lab required me to standardize the base, or sodium hydroxide. And to standardize this base, I performed a separate titration. This essentially confirms the base's concentration, which makes future calculations more accurate. And now that we have a standardized base, we can start titrating into the analyte. In this case, the analyte is a mixture of dissolved orange-flavored baby aspirin in ethanol with some drops of phenolphthalein. I slowly added the sodium hydroxide until the analyte turned faint pink. This indicates that the amount of base added to the analyte is the same as the acid in the analyte and that the pH is neutral. So after running multiple trials and averaging my data, as you just saw, I found that the baby aspirin was only about 40% aspirin pure. Without titrating baby aspirin, scientists wouldn't know the purity, and with other calculations, even the pH of it. This is especially important to pharmaceuticals and medicine because by not achieving the correct pH, the medicine may not be stable, effective, or even soluble for the patient. For example, Tums are effective because they are a basic substance. If they were not basic, it wouldn't prevent heartburn and indigestion in the acidic environment that is the stomach. And in other examples, it could even be unstable and harmful to us. Overall, knowing the process of titration is very important for both accurate results and to make sure that the purpose of the drug is fulfilled.